Assalamu alaikum. My name is Rabia Asad and my roll number is EP1770119. My topic is tea maize and the portion which I explain here is animal behavior. As we know that when animal is placed on any apparatus, it will experience the behavior, it will show some behavior. Here I would like to explain the evaluation of the spatial memory by using the reward alternation procedure and it will be carried out in a tea maze apparatus. As a tea maze, it is enclosed apparatus. It is placed horizontally. The mouse is placed at the base of the tea, and it contains the two arms called the goal arms, which contain the food rewards. In this task, two trial run was given to the mice. In the sample arm, there is a door that is closed that confine the mouse to a specific arm or probe the mouse to enter to only the one arm. So there is the two common protocol to assess the tea maze, that is the spatial memory and the learning memory. Here the mice is placed in maze prior to test. Rewards are placed at both the two ends. One door is closed and the one door is closed and the mouse moves to the open arm and consumes the reward. The door which is in the sample one is now then open and the mouse in return to the start. It is now choose that which arm he previously explored or visited. If it is choose correctly, it consume the reward, and if it choose incorrectly, it will see that food wall is empty. Thus, the accurate performance depends on remembering which arm was visited most recently during the sample one in order to guide the appropriate responding in the choice run with the correct spatial response varying from one trial to the next trial. Rodents, especially with the hippocampus lesion, it display the chance level of the performance on this task, and the glucose A mice also impaired, and the infusion of the AP5, which is the NMDA receptor antagonist, into the dorsal hippocampus will also affect the spatial working performance in the mice. So by using the tea maze, we can assay the learning and the spatial working performance in mice. Now I would like to share some information through article regarding the behavior of the animal. As we know that hippocampus play an important role in the memory and learning. So the dorsal hippocampus has been implicated in the spatial memory performance. Therefore, any lesions in the dorsal hippocampus, it may affect or changes the brain structure. And the infusion of the NMDAR antagonist, that is the DAP5, into the dorsal hippocampus, it may also impair the spatial working memory performance. Now moving toward the introduction, here the two experiments will be carried out. In experiment 1, there is a complete hippocampal lesion that produce robust and the lasting impairment on the spatial memory task, and it will indirectly affects the brain part rather than the cell loss in the target region. So the infusion of the musky mole, as the musky mole is an psychoactive constituent, it is a related species of the mushrooms, and it is a agonist of the GABA neurotransmitters. So we compare the studies. So we compare the effects of the dorsal and the ventral hippocampal infusion of the musky mole on spatial learning using the spatial working memory, and therefore we compare the dose response function for musky mole infusions into both hippocampus subregion. Now in the second experiment, the importance of the NMDAR function, that is the intracerebroventricular infusion of the antagonist of the NMDR, that is the AP5, resulted in delay related impairment in choice accuracy on the same spatial as this route result in an MDAR blockage in numerous brain regions and not just the hippocampus. Now moving towards a method, and as we took 3 male rats, and the apparatus which we use here is the T-maze apparatus. In this method, in both the experiment, the rat were trained pre-operatively. As the rat will spend, or their habitual period is for the 5 days. For the first 3 days, rat were placed on the maze in pairs and left to explore and collect the food rewards for 10 minutes. During the day 5 and day 4, the rat were placed individually on the maze and allowed to explore freely for 5 minutes. So all the rats ate food from the food well, and the end of the arm training on the rewarded alternation task then followed, which I previously explained that each trial has given a 2 runs. There is the goal arm is blocked, allowing the rat to enter only the one goal arm, whereas and to receive the food pallet, whereas in the second run, the block was removed and the rat was placed on the maze and giving a free choice to enter either the arm, that is a goal arm, or the sample arm. Rat received 2 pellets for choosing the previously unvisited arm. Choosing the arm previously visited or explored in the sample run yield no reward. So the time sample is noted between the sample and the choice run was approximately the 10 seconds. Now the drug that is in experiment 1, muscimol, was initially dissolved in the normal saline at the concentration of 1 mg per ml, and the stock is diluted to create an individual aliquots at 4 different concentration, which is then frozen, and these salines serve as a vehicle, that is the administration of this vehicle into the rat and see the behaviors of the mice. In the experiment 2, muscimol along with AP5, which were dissolved in PBS, and it served as a vehicle and will give in the experiment 2 rats. Now we see the result of both the experiment in the mice, and it will shown that post-operative performance was slightly better than the pre-operative performance in both the group. As we know that muscimol infusion of the muscimol in both the experiment, as the infusion of the muscimol in both the experiment of the mice, it will improve, or it will improve the spatial working memory performance. As we know that muscimol is an agonist of the GABA neurotransmitter, and the GABA neurotransmitter is an inhibitory neurotransmitter in the brain and play an important role in the working and in the memory and learning. Therefore, their performance will be improved by the infusion of the muscimol drug.